Hello everyone, this is Aloysius Guy in a new tutorial, and today's topic is DNA replication. So we will cover how DNA replication works, and we will understand what components are involved in this process. But before we start, please try to check the time codes in the description below so you can go directly to the chapter that you're interested in. Let's start. So before we start understanding what is the DNA replication, we need to have a quick look at the structure of the DNA and the DNA double helix. So as we all know that within our cells, they have a nucleus, okay? And within the nucleus, there is the DNA or the genetic material. Our DNA is packed into very packed structures called chromosomes, and if we unpack these chromosomes, we can find the DNA. So the DNA is made up, if we look at the structure here, it's made up of a sugar phosphate backbone with nucleobases, okay? So the sugar phosphate backbone with a nucleobase, they are together called nucleotide. So the building blocks of our DNA are 4 different nucleotides: adenine, guanine, thymine, and cytosine. So you might be asking yourself how these different nucleotides, how the interaction between these different nucleotides allow the DNA to maintain its double helix structure, okay? And the answer for that is the formation of hydrogen bonds between these nucleotides, okay? So this double helix structure is maintained because of the hydrogen bonds formed between nucleotide. So let's have a look or a closer look at that, okay, at the DNA structure. So this is the sugar phosphate backbone. So this is the sugar group and this is the phosphate group, okay? And what we have in the middle is the nucleobases, okay? And these dotted lines here are representing the hydrogen bonds. And as we can see that between adenine and thymine, there are two hydrogen bonds, and between cytosine and guanine, there are three hydrogen bonds. The presence of these hydrogen bonds is essential to maintain this double helix structure. So before we get into the process of DNA replication, we need to understand first why is it important. And the main reason and the main importance of DNA replication is that it ensures the transmission of genetic information from one generation to the other. What do I mean with that? And that's what I mean. So when we have a parental cell here, okay, so a parental cell will undergo cell division to produce 2 daughter cells, okay? So this is a parental cell and these are called daughter cells, okay? So as you can see here, the DNA components during cell division will be duplicated within the parental cell, and then each daughter cell will have an identical copy of the DNA identical to the parental cell, okay? This process is called the cell cycle, okay? And what we are interested today in this tutorial, we are interested in this stage of cell cycle, which is called the interphase, because this is the stage where DNA replication occurs, okay? Which means that this is the stage where the parental cell will duplicate its DNA information or DNA genetic material, and then will be splitted into 2 new daughter cells. You need to remember that because of DNA replications, when somatic cells divide, all your body would have identical DNA information. That's why it's very crucial for healthy development of our bodies. So now we need to get introduced to the components that are involved in replicating our DNAs. And what we need is a template DNA, a nucleotides, and then we need enzymes and other proteins. So these are the 3 main components that are important for carrying out DNA replication. So when we say DNA templates, here we are referring to the parental cell DNA, okay? So that's the parental cell DNA that we need to copy from. Then we need nucleotides, which are important for synthesizing the new strands that are going to be copied into the daughter cells. And then proteins and enzymes are involved in unwinding this double helix structure, okay? And also with carrying out the addition of nucleotides and synthesizing the new strand. So now let's start with the DNA replication process. And so the process starts with the first stage, which is called the initiation stage. And this stage, what happens mainly in this stage is the recognition of an area of the DNA called the ORI, or the origin of replication. So what is this origin of replication area? Okay, so here we have the DNA, and within the DNA, there are very specific sites from which DNA replication starts. So DNA replication does not start randomly from all over the DNA. However, but instead, they start very specifically at very specific sites, and these sites are called origin of replication, or the ORI. So these sites have very specific DNA sequences. Also, they have very specific structural features that are identified by a complex protein called the ORC. So ORC is shortened for the origin recognition complex. So this complex its main function is to identify these areas from which the DNA replication starts. Okay. This protein complex, the ORC, has 6 different subunits, and every subunit would have a very specialized function that is involved in the initiation of DNA replication. So what happens is that once the ORC complex binds to the origin of replication, what happens next is that this protein will start recruiting other different proteins that are involved in the initiation of DNA replication. And 2 of the main proteins that are recruited are CDT1 and CDC6. So what happens then is that the protein, the ORC complex with the recruited proteins will result in forming a small opening within the DNA double helix, or the DNA double strand. Okay, this small opening will be recognized by an enzyme called helicase. So we will go further into more details about the role of helicase. But what we're going to show here is that one form of helicase is called the MCM, which is one important helicase found in a human. Okay, it's made up of 6 different proteins, and they are assembled, as we can see here, in a hexameric ring structure. Okay. So now let's have a look at the second stage of DNA replication, which is DNA unwinding. And the main thing that happens within this stage of DNA replication is the creation of what we call a replication bubble. So what is this replication bubble that we are talking about? So as we can see here, this is the helicase that we just showed you. And as we can see, that one helicase will bind to this band, to this strand, and one helicase will bind to this strand of the DNA. And this is called the replication bubble. Okay, so what does the helicase do is that they are responsible of breaking the hydrogen bonds between the nucleotides of the DNA, which results in unwinding this double helix structure and producing this bubble. And as we showed, if you remember at the beginning of this tutorial, that adenine and thymine, they have 2 hydrogen bonds. Guanine and cytosine, they have 3 hydrogen bonds. And these bonds are broken by the effect of helicases. Okay. A very important thing that we need to mention is that the direction or the movement of helicase during DNA replication, and you need to pay very close attention to the movement and the direction of movement. So this helicase will move from the 5' to the 3', and that helicase will move from the 5' to the 3'. So what will this result in? It will lead to making the replication bubble even larger, because this helicase will continue moving in that direction, and this helicase will continue moving in that direction, making the replication bubble much larger, okay, and an increase in size. Also, we need to mention here that, as we can see here, these 2 Y shapes that are within the replication bubble, these Y shapes are called the replication fork, okay? And every replication bubble would have 2 replication forks. just bear in mind these terms and these words, because during this process of DNA replication, we will come back to them. So I want you to understand when I say replication fork, what I mean with that, and when I say replication bubble, what I mean with that. So the replication bubble has two replication forks. Maybe you're asking, what are these structures that are added to this image? So these structures are called topoisomerase, and they are proteins that are very important in DNA replication because they are preventing the buildup of tension that occurs due to the unwinding of DNA. And because as you unwind the DNA, if topoisomerase is not there, maybe knotting will start forming within the DNA. So topoisomerase will relieve this tension and allow the DNA replication process moves smoothly. Now we're going to have a look at another component that is present within the replication bubble, and it has a very important role that is playing. So as we can see, this is the replication bubble, and we have here three proteins, and these proteins are called the SSB, or the single-stranded binding proteins. So what is the role of these proteins, and why do they bind to the replication bubble? So the main function of these proteins is that they bind to the single-stranded DNA. So as we can see here, the DNA is double-stranded. However, within the replication bubble, the DNA is single-stranded because these two are separated from each other. So if these proteins did not bind to the single-stranded DNA, what happens is that these two strands will just rejoin together, because that's the tendency of the DNA. The DNA always tends to rebind to form this double-strand structure. So this protein prevents the rebinding of these two strands. Also prevents the degradation of these single-stranded DNA, because sometimes nucleases might bind to these single-stranded DNAs and degrade them. However, these proteins protects the replication bubble and the single-stranded DNAs from getting degraded. If you look also within the replication bubble, we have these arrows now. So in these arrows, I am referring to the synthesis direction of the newly formed DNA strand. So as we can see, the top, we have the top template and the bottom template. Okay, so for each template, you need to understand this: the forming of the new DNA strand must be anti-parallel to the template. So this template moves from the 5 direction to the 3 direction, okay, to the 3 prime. So therefore, the synthesis of the new strand will be in the opposite direction. So the 5 prime to the 3 prime. It's going to be in that way. And the same thing for the bottom templates. So the bottom templates also, it's going to be in the opposite side. So this is 5 prime to 3 prime. So here it's going to be the 5 prime on the new strand to the 3 prime on the new strand here. So the question now is, why do the DNA synthesis always occur on the 5 prime to the 3 prime? So you need to understand what does the 5 prime mean and the 3 prime means to understand why DNA synthesis only occurs in that direction. And that's what we're going to cover now. Okay, so what do DNA 5 prime and 3 prime refer to? Before we answering that, we need to go back first to the structure of the nucleotide. So as we can see here, so the nucleotide is made up of a nitrogenous base, which is adenine, thymine, guanine, cytosine, and then a sugar group, and then a phosphate group. Okay, and so these are the 3 main components that makes the nucleotide. As we can see here, within the sugar group, there are 5 atoms, 5 carbon atoms, and every angle is a representation of a carbon atom. So the first, the second, the third, the fourth, and the 5th carbon atom. Okay, and that's what we mean with the 5th prime and the 3 prime. So they are referring to the number of the carbon atom within the sugar group in the nucleotide. So when we say a 5 prime, we are referring to the 5th carbon atom that is bound to the phosphate group, and when we see the 3rd prime or the 3 prime, we are referring to the third carbon atom that is binding to the hydroxyl group. You need to remember that. So the 5th prime is bound to the phosphate group. The third carbon is bound to the hydroxyl group. Okay. So here we have 2 DNA strands. So this strand runs from 5th prime to the 3 prime, and this strand runs in the opposite direction from the 5th prime or the 5 prime to the 3 prime. Okay, and these are the hydrogen bonds between the nucleobases. So as you can see here, when I say 5th prime, I here refer to the end of the DNA strand where there is a phosphate group. However, when I say that this is a 3 prime, then we are referring to the end of a DNA strand that ends with a hydroxyl group. Okay, so you need to remember that because this is very important to understand DNA replication. 5th prime, phosphate group, 3rd prime is the hydroxyl group. So then the question is now, why the synthesis is always moves from the 5th prime to the from 5 prime to the 3 prime? And the answer for that is, so if we look here at this strand, this is an ongoing synthesizing strand that is being synthesized. And as we can see here, this is the 5 prime, which is the phosphate group. And here is the 3rd carbon, which is bound to the hydroxyl group. This is the new nucleotide. So when a new nucleotide wanna bind to the previous nucleotide, what happens is that the phosphate group on the new nucleotide will bind to the hydroxyl group on the 3 end on the 3rd carbon in the previous nucleotide, forming a covalent bond which is called the phosphodiester bond. And that's what governs the movement of the synthesis of the DNA in just one direction. So only the 3rd prime can accept new nucleotides, and they can bind to. So nucleotides cannot bind to that end. So that's in a nutshell the whole process of what does 5 prime and 3 prime mean. So now we go back to the DNA replication and the replication bubble. So the last thing that I need to mention in the second stage of DNA replication is that if we look here within the DNA, within the replication bubble, we can see here a single-stranded DNA and here a single-stranded DNA. So the thing that you need to understand is that the enzyme that is responsible of making the new DNA strand, which is called DNA polymerase, cannot bind to this naked single strands of DNA. And therefore here where it comes the importance of an enzyme called primase. So what is primase? So primase allows the DNA replication to enter the 3rd stage, which is the DNA elongation stage, okay, which is the stage where now we start synthesizing the new strand. So how is that? And how primase plays a role on that. So this is the replication bubble, and as we can see that these single strands are naked, so DNA polymerase cannot bind to these strands. Therefore, the role of primase is that primase enzyme can bind and recognize the specific sequence found within the origin of replication, okay? And what they would do is that they will synthesize short RNA primers within the origin of replication on each strand. So as we can see here, that this primase bound to the origin of replication and then synthesized a small short RNA, and the same thing happens on that strand. And the thing that you need to remember is that the primer on that strand will have the 5th prime here and the 3rd prime here, which means that the elongation is going to be in that direction. However, on this strand, the primer, this is the 5th prime and this is the 3rd prime, so the elongation is going to be in that direction. And that's why primase is so essential to start DNA replication, because since now we have primers here, now the DNA polymerase can bind and start synthesizing the new DNA strand. So now we need to get to the stage where we need to understand what is the role of DNA polymerase in the DNA elongation during DNA replication. So DNA polymerases are enzymes that are responsible of synthesizing new DNA strands by adding one nucleotide after the other, as we showed previously. Okay, so within the replication bubble, we have here the two origin of replications, and then this primer and this primer, which were synthesized by Primase. And once this primer is synthesized, DNA polymerase will identify this region, and what happens is that we'll start synthesizing the new DNA strand in that direction. So this is DNA polymerase, and this is the second DNA polymerase. And as we can see that we have two different polymerases in the human, and the first polymerase is called polymerase epsilon, and the second one is polymerase delta. And the main difference between these two is that this one synthesizes a strand called leading strand, and polymerase delta is synthesizing a strand called the lagging strand. We will understand later what do these terms mean. The second thing that you need to pay attention to is that DNA polymerase, as we explained before, it adds nucleotides in that direction, 5 to 3, and the same thing here, 5 to 3. The second thing here is that this protein, which is the proliferating cell nuclear antigen, or shortened for pCNA, and it has two main functions. So the first function is that it stabilizes the whole DNA polymerase binding to the DNA, so it allows binding. And the second role is that it allows the DNA polymerase to synthesize a long DNA strand without the need to dissociate from the DNA strand. That's why this protein is also very important for DNA for DNA replication, because it stabilizes the polymerase. So now the question that we need to answer is, what are leading and lagging strands? What are these strands, and why do they have different names, and why different polymerases are synthesizing these strands? So now we will start with covering what is the leading strand. Okay, so here we have the replication bubble, and as we can see that the DNA polymerase, what it's doing here is that it's copying this template into this strand, and copying this template into this new strand. Okay, and that's what the DNA polymerase is doing at this stage. So pardon my drawing is not perfect, but I'm just here showing, trying to get you the to understand the idea. And so if you look here, so this is the helicase moves in that direction, and this is the second helicase moves in that direction. And as the helicases are moving in those directions, the replication bubble will get larger. Okay, so what happens is that the DNA polymerase will keep synthesizing this strand in that direction, and this polymerase will keep synthesizing the other strand in that direction. So these two strands are called the leading strands. So what are the main characteristics of the leading strand? So there are two main features of leading strands. So the first one is that these strands are synthesized continuously without any interruption. And that's because they are moving in the same direction as the helicase. So this helicase is moving in that direction. So for example here, this helicase, it was a short template strand, then it moves further as we show here. So new template now is free, and then the DNA polymerase will start synthesizing and copying that strand as well. The same thing happens here. So this DNA polymerase will keep synthesizing all the newly exposed DNA strands in the template as the helicase is moving in that direction. Okay, so if those templates are copied by the DNA polymerase, so the question is, how about these templates? How DNA polymerase is gonna copy them? Because this polymerase will just keep moving on that strand, and this polymerase will keep moving in that strand. However, this area behind it, how is it going to be synthesized? And there where it gets to the lagging strand. So now we get to the lagging strand. And what is the lagging strand? So as we can see here within the replication bubble, now we know that this strand that is going on the same direction as this helicase, this is called the leading strand, and this strand is also called the leading strand. So, and now we mentioned that these two free areas will be synthesized as well. However, their synthesis is a bit different. So what happens is that as this DNA template will be naked, the primase will also bind. After binding to the origin of replication and starting the DNA replication, primase will bind to another naked DNA and then synthesize a short RNA primer. Then DNA polymerase will be able to bind to the DNA primer and start synthesizing the DNA strand, the new DNA strand. And this synthesis will be continuous until it reaches the primer of the previous DNA strand. The same thing happens here. So when we have a naked DNA strand, the primase will bind, make a new primer, then DNA polymerase will be able to identify this primer and then start synthesizing the new DNA strand until it reaches the primer of the previous DNA strand. So as the replication bubble gets larger, because the helicase moves in those two directions, okay, what happens is that this strand was this strand, but now the replication bubble gets larger, so now we have a new template that is exposed. So what happens is that the primase will also bind to that new DNA strand, and then the DNA polymerase will bind as well, and then start synthesizing the new strand until they reach the primer of the previous strand. And the same thing happens here as well. So, and all these syntheses, they are in the 5 to 3 direction, and as well in the 5 to 3 direction. So these short fragments that we can see here, these are called the Okazaki fragments, okay? And multiple Okazaki fragments are known as the lagging strand. So the main features or characteristics of a lagging strand is that it's made discontinuously. So it's not like the leading strand where it's going to be continuous because they are in the same direction as the helicase. However, these strands will stop after they reach the 5' of the previous RNA primer. That's why they are made in small fragments. So now the question is that since the lagging strand is made up of short fragments, how these DNA fragments will be joined together so at the end we would have a full continuous DNA, and that's what we're going to see now. So joining the Okazaki fragments within the lagging strands is a very critical stage at DNA replication. And it mainly occurs through 3 different stages. So the first stage involves an enzyme called RNase H. So RNase H will bind to the primers, okay? So as we said that there are primers that were synthesized by primase. So the role of RNase H is to remove the nucleotides that are making that RNA primer, okay? Once these primers are removed, what happens is that DNA polymerase, and as we said before, that polymerase delta is the one responsible of making the lagging strands, will make a short DNA strand that replaces the RNA primer that was removed by RNase H, okay? Once that small DNA was made, so now this strand will be joined to that strand, and the newly synthesized DNA will be joined together using an enzyme called DNA ligase. So DNA ligase is responsible of making this continuous strand that used to be two strands, two Okazaki fragments. And these are the three main stages of how getting a continuous lagging strand after the formation of Okazaki fragments. So now we get to the last stage of DNA replication, which is the termination. So the termination occurs when two replication forks meet. So because the replication forks progress along the DNA strands in two opposite directions, so when these forks meet each other, this signify a signal that the DNA between these two replication forks was copied and the process of DNA replication has ended. And this will lead and or signify to the cell that now it's ready to move to the next stage of the cell cycle after the interphase. That's why termination is so important, and all the proteins involved in that recognitions are so essential because it allows the cell to be aware or realize that the DNA was copied, and now to move further through the cell cycle. And now here, as usual, some questions that are exam-like questions that will help you test your understanding of today's tutorial. And with that, we reach to the end of this tutorial. Thanks for watching, and please don't forget to like, subscribe, and comment, and also turn on the notification button so you can get all notified of all our new videos. Also, please check the time codes in the description below so you can go very quickly to the chapter that you're interested in. Thanks for watching.